differential leukocyte count is about counting the different types of leukocytes. So this is, you write, you know like this, that after preparation and staining of a blood smear, identification of different types of blood cells under the microscope, and then differential leukocyte count. From the examination point of view, that question you will have to face, you have to do this experiment. If there are 10 marks allotted, you have to do this for 10 marks. One of the topics is this differential leukocyte count. So preparation and staining of blood smear, identification of cells, and differential leukocyte count. Then there are two topics, total erythrocyte count and total leukocyte count. These are two separate topics under hematology. Total leukocyte count. So if you have to write properly, then preparation of bloods and staining of blood smear, that will be preceding differential leukocyte count. Okay, without preparing the smear, you can't do the count. So first it will be preparation and staining of blood smear, followed by learn to identify the cells, and then do the differential count. Okay. So total leukocyte count, total erythrocyte count, these are two separate topics. Then estimation of hemoglobin. All these topics you will find among the competencies which is mentioned in the CBME 2024, estimation of hemoglobin. Then estimation of bleeding time and clotting time. Those who are there in the last week of October already have written these down or already know this by now. Estimation of hemoglobin, estimation of bleeding time and clotting time, determination of blood groups. So these are the topics which you are to perform yourself during the exam, and marks will be allotted accordingly. Okay. So three topics are there: differential leukocyte count, total erythrocyte count, total leukocyte count. This takes a longer time, considerably longer time. Usually these, if these are there, one of these is there in the question, then you have to conduct one experiment, but for the rest they are shorter experiment, and there will be the hematologist practical examination session is roughly 1 hour, 45 minutes to 1 hour. So you have to complete your experiment and report to the examiner by that 1 hour. So in that 1 hour, this, if you have one of these three topics which I told at the beginning, differential leukocyte count, total erythrocyte count, total leukocyte count, one of these, then only one experiment is usually allotted for, and 10 marks is that, the full marks is 10 for that, the whole hematology 10 marks is for that experiment, but for the rest ones there may be a combination. If one, where 2 or 3 of those, you have to estimation of hemoglobin, blood groups, bleeding time and clotting time, 2 or 3 of those you have to you have to perform. Okay, that you will get acquainted during these internal assessments, how to face the exam. So during these internal assessments, again you will be told. So these are the topics for hematology. We had also mentioned about the topics in clinical physiology. Amphibian physiology is a brief part of the syllabus. What are the topics in clinical physiology? Just in the last class with you, I discussed it on that Tuesday. Before the last Tuesday, there was no class, and those who are new will get acquainted with it. We will go to today's topic now. Okay, we will get acquainted with it. Just know from your colleagues, clinical physiology, there are a lot of topics. We have discussed only one topic till now, that is examination of the arterial pulse. We have discussed, and that will be done for those who are joining, they will get to know about it. For now, the topics in hematology which we have discussed, one class was there on the compound, on this microscope, study of the microscope, and preparation and staining has been done. So we will learn to identify the cells, but for those who have joined newly, I am just telling you how to prepare and stain a blood smear. Okay, so we'll go to the laboratory. There will be slides. So there will be slides, you have to take glass slides, and on one glass slide, those who are new, please take the help of them who are already done the class, and we will be there to assist you. With the glass slide, you have to take a drop of blood. You have to take a drop of blood on a glass slide. Now, how to collect this drop? So, in hematology, you should learn how to collect capillary blood. Always you may not be supplied with venous blood, and some experiments are there, hematological experiments, which cannot be done with, which you are not to do with venous blood, you have to do with capillary blood. So what is the difference between capillary blood and venous blood? Blood drawn from veins, if you have undergone, anytime you have any of you donated blood? Okay, and maybe most of us have to undergo blood test for for when we are diseased. Okay, so drawing that blood, usually it is done from a vein in the cubital fossa. The cubital fossa is this. When you just flex, this is the flexion movement of the elbow. There is the cubital fossa is in the front of the elbow. That is area is called the cubital fossa. So from there, from the cubital vein, blood is drawn and collected for blood evaluation of the blood in the laboratory. So that is, the blood is drawn from the vein. But capillary blood, how to collect? Capillary blood is collected from by pricking with a needle from some areas. Usually it is collected from the finger. So it may be collected from the ear lobe. Okay, in children it may be collected from the heels. Okay, so it is usually collected from the fingers. Sorry. So you are to use a needle, and for the beginners you have to prick your own finger and collect the blood, drop of blood on a slide. So this, how to prick the finger, there are some precautions to be followed. One thing you have to know, the prick has to be a single prick and a deep prick, otherwise you will not be able to collect blood. So if it is not a single and a deep prick, then you will not be able to collect blood properly. If it is a very superficial prick, after even before one drop of blood comes out, the blood flow may stop, and then what usually is the mistake, that if you try to squeeze the blood out of the finger just after a single prick, just try to squeeze the blood, then instead of blood, tissue fluid will come out. So fluid which is interspersed between the cells, that is called tissue fluid or interstitial fluid. Now interstitial fluid will not help in your experiments with blood. Okay, there is nothing. Why are you laughing? Is there anything to laugh? So interstitial fluid will not help in your experiments with blood. Blood mixed with interstitial fluids, the experimental results will also go wrong. So single deep prick, you have to do a single and deep prick and then put that blood on a glass slide, and with another glass slide you have to make the blood smear. Now for that there is a that means you have to follow the procedure stepwise. That is, you have to take the blood from the finger. So before that there are precautions to prick the finger and take the blood. What are the precautions to be taken? That is, or what is the procedure? So procedure means, don't straight away take a needle and prick the finger. Just first clean the area. So use a piece of cotton and spirit which will be supplied, and just clean the area. Okay, clean the area with the help of cotton and spirit. Allow that area to dry. So usually we do it from the middle finger or the ring finger. So we allow it to dry. Okay, so what is the purpose of allowing it to dry? Because without drying, again, if that liquid which are using for cleaning the finger, that may get mixed with the blood if it is not allowed to dry. But the main reason is this, and the what is the purpose of using this liquid to clean, to disinfect the area? So this disinfection requires some time, maybe 1 minute. So as you allow it to dry, that disinfection occurs. So it takes, if you straight away prick the finger after just rubbing the liquid, the spirit to your finger, then that time is not given, that time is not being allowed. So disinfection of the finger plus, so that blood does not get mixed with whatever small amount of fluid is there, to prevent that, we allow it to dry and then this carry out this pricking. So after pricking, blood will come out. You have to just, without squeezing the finger, the blood should flow out freely, and put 1 drop or maybe 2 drops on 2 separate slides. Okay. And then another glass slide you have to hold with your dominant hand. That means usually most of us are right-handed. If you are a left-handed person, then use the left hand. So another slide you hold at an angle of 45 degree with the first slide. The first slide on which the blood is there, you have taken the blood drop, is on a horizontal surface. So we use another slide and put it in front, just in front of the, not directly over the blood drop, but in front of the blood drop. Now slowly, now after that, you just imagine it, we will do it in the laboratory, you just imagine it on a piece, on a glass slide there is 1 drop of blood. We take another glass slide, hold it at an angle of more or less 45 degrees. That will just do that by your whatever your own senses, that it is at 45 degrees, you put it in front of the drop and draw the slide backwards, so that the slide which you are using with your hand, it is in your hand, that is that slide, there is an edge of the slide is on the other slide on which the blood drop is there. So the blood spreads according along that edge. Okay. And then you draw the, just with 1 single stroke, draw the smear on the slide on which the blood is there. Okay. So it will be somewhat like this. This one slide will be placed on a horizontal surface. The blood drop maybe you have taken here. So blood drop is to be taken near one end of the slide, not in the middle, but near one end of the slide. And then you will put the slide which you have taken with your other hand and put it just in front of the, at an angle of 45 degrees, like you have put this at an angle of 45 degrees, you put this slide and then just draw it backwards, so that over this edge, this blood drop now is spread along this edge. And now you make a single stroke on this slide and draw this smear. The blood will spread along this portion. Okay, so that will make the blood smear. Now ideally a blood smear is, ideally what will happen, as you draw, the blood will cover, here it will, blood will cover almost the whole breadth of the slide. Then it will cover somewhat less over this area, then it will cover somewhat less of the breadth. An ideal smear is usually like this, it is called, it becomes like this, it becomes a tongue shaped smear. Ideally it is a tongue shaped smear. If it is a straight one also, that will you can work with that, but ideally it should be a tongue shaped smear. Okay. So after this spreading of the blood drop, making the blood smear, you have to stain this blood smear. Now this staining is done here, what we use with staining is done with something which is called the Leishman stain. Those who have attended the previous class know this, it is done with the Leishman stain. So after drawing this smear, just wait for say 1 minute and apply Leishman stain on this smear. Now how to apply Leishman stain? Don't apply Leishman stain as you wish, don't do it apply the Leishman stain drop by drop. There are arrangements for that. You have to apply Leishman stain such that it just covers the smear. So in your initial days, you will all the stains smears which we make may not be tongue shaped. It may be, say, if it has become a, just you see wherever the stain is and then just put the stain over that portion only on which the blood smear you have drawn. Don't make it, if the blood smear, if it is up to this, then don't put stain over this area. But during putting the stain over the blood smear, just count the drops you are using. So there is no hard and fast rule that it should be 6 drops or 10 drops or 12 drops or 1 drop or 2 drop. So usually it is 4 to 6 drops. Okay, if you make a good smear, it should be 4 to 6 drops. So just count the number of drops. Okay, that will be needed in after this, just at a later stage, that will be needed. So count the number of drops. Say you need 5 drops to cover that blood smear with the Leishman stain. So after putting this Leishman stain, wait for 2 minutes. Okay, 1.5 to 1 minute, 2 minutes. Okay, after putting this Leishman stain, count the drops and putting this Leishman stain, wait for 2 minutes. After waiting this, at this stage, you are to put distilled water over that smear. You have put the Leishman stain on the smear. The Leishman stain is of say deep purple color. Okay, now you put distilled water. Distilled water, you put distilled water, or you may use buffered water. Now distilled water, the rule is that if you have put say 5 drops of Leishman stain, then put 10 drops of distilled water, that is double the amount. If we use buffered water, it is the same amount as that of the Leishman stain. So that is the reason for counting the drops of the Leishman stain, so that you know how much distilled water or buffered water we have to use. If it is distilled water and you have put say 6 drops of Leishman stain, then you put 12 drops of distilled water, because after putting the Leishman stain, it is not possible to visualize the smear as such. The stain will be spread over the smear. So now how you put the distilled water, you just recall how many drops of Leishman stain I had used 2 minutes earlier. So that is maybe 5 drops, maybe 6 drops, and just double the amount of distilled water you use. After you put this distilled water, if it is buffered water, the same number of drops have the Leishman stain. Now after you put this distilled water or buffered water, you have to wait for 10 minutes. Okay, so this earlier 2 minutes which was required after the application of the stain, that is called fixation time. The smear is getting this, along with this, the smear is getting fixed. So all these what is present, that is getting fixed to the slide. That time is being taken, and after the 10 minutes after putting the distilled water, that is actually the staining time, when the blood cells, the components of the blood are being stained by so that it is, the purpose of staining is to identify the cells under the microscope. So that time is the staining time. Okay, those 10 minutes. Now after 10 minutes are over, it is 10 minutes. 8 to 10 minutes are over. So you have to just, whatever excess is there, you have to just wash it out gently, the slide under tap water. It is advisable to do it under tap water, but it has to be very careful. Okay, so what you do is hold the slide. You don't have to write anything, it is all in you have to follow a physiology practical textbook, and everything is there in the books, and while you do it, you will learn it. There is nothing to write down. Okay, so you just under the tap water, if you hold gently, very gently, the tap water has to be allowed to fall very gently over which end of the slide? That end of the slide where initially the blood drop was taken, and it is just the excess fluid, whatever is there, is washed away. But don't do it don't use the tap, don't allow the water to run very fast, otherwise it may wash away everything. So it will be very gentle, and more advisable is that you just hold the slide where the blood drop was there initially, you just put your thumb over there and allow the water actually to fall on your thumb very gently, and then it is washed away, not directly on the slide. If you are very careful, even if it is directly on the slide, it may be, but it is your purpose may be served, but still you put your thumb and allow the water to fall on your thumb, and then it will wash away. Okay. Now, what happens after 10 minutes? It looks like somewhat greenish. It is called a greenish scum. It is somewhat greenish scum is there. That is washed away, and after washing away, the smear has been fixed. Allow it to dry. After drying, you will see it under the microscope. So this is the process of preparing and staining the smear, and then you have to look for cells under the microscope. Okay. No. After you will find that greenish tinge once those 5-6 minutes are over, but you wait for 10 minutes and allow sufficient time to stain, and then wash it, and then you will. Yes, the only will find the smear over the slide, and you put the slide under the microscope, and you have to look for cells. Now, how cells are identified? What are the most of the cells in a what are the blood cells? RBCs, the normal blood cells are RBCs, WBCs and platelets. Although platelets are not whole cells, they are cell fragments, and these are called formed elements of blood. These will be discussed in hematological theoretical classes. Formed elements of blood, RBCs, WBCs, red blood cells or the erythrocytes, white blood cells or the leukocytes, and the platelets. So usually it is for the in the initial stages, it is sufficient to identify the RBCs and the WBCs. RBCs are huge in number compared to WBCs and platelets. The theoretically we know the total count. What is the total count of RBCs in blood? We express it in millimeter cube of blood. Per millimeter cube of blood, we express the figure. It is about 4.5 to 5.5 millions, around 5 millions of RBCs, whereas WBCs are only 4,000 to 8,000. 4,000 in standard textbooks, it is written 4,000 to 11,000 per millimeter cube of blood. Okay. Again, per millimeter cube of platelets are like 1.5 lakhs to 4 lakhs. Okay. Say 2-3 lakhs like this. So these are the normal ranges of these counts. So you can see RBCs are huge in number compared to WBCs. Staining by the Leishman stain, RBCs will appear as pinky structures without the nucleus. Okay. Now just before why the RBCs will find them as pinky structures. So Leishman stain contain Eosin and Methylene Blue as the staining components. Eosin, Eosin and Methylene Blue. So Eosin stains the cytoplasm of the RBCs and the granules of eosinophils. Okay. Eosinophils are a type of white blood cells. The white blood cells are broadly of two types, if we consider like the granulocytes and the agranulocytes. So the granulocytes are containing granules. Okay. In case of the granulocytes are named as neutrophil, eosinophil and basophil. Agranulocytes are named as lymphocyte and monocyte. Okay. So these granulocytes, eosinophils, basophils and neutrophils, as from the name granulocytes, they contain, that's why they are named like that. These contain granules. In case of the eosinophils, the granules are more coarse, and in case of neutrophil and basophil, neutrophils are most fine granules, basophil may be intermediate between these two. Eosinophils, and when you look under the microscope, the granules are coarse. Okay, and these granules of eosinophils stain like that, are stained by eosin, the component eosin of this Leishman stain. And what else does the eosin stain? That is the cytoplasm of the RBCs. Now the RBCs do not contain any nucleus, so you will find these as round pinky structures. Although actually the shape of the RBCs is biconcave shape, we will learn about that, but it is round pink. You will, under the microscope, these appear as these round pinky structures, and most of the cells which you will find at the microscope are RBCs. You will find a lot of these structures, these round pinkish looking structures, and among them in few areas you will find the WBCs and few platelets also. But most of the cells which are looking like this, if in a well stained smear you have followed the procedure of staining properly, then you will find most of the cells, this round pinky structure, those are the RBCs. Okay, and these will look pinkish. Now, how to know about the WBCs? WBCs mostly just by first look. It is differentiated from the, if it is a well stained mirror, if it is very easily differentiated from the RBCs. WBCs are not like those pinky structures, and WBCs, whether it is a neutrophil or a eosinophil, basophil, or a lymphocyte or a monocyte, whatever, these will look distinctly, that means distinctly, clearly it can be differentiated from the RBCs. And what part of the stains the nucleus of these WBCs? All the nucleus of all WBCs are stained by methylene blue, even the granules of neutrophils and basophils. So these will look, I may call it deep purple, okay, nucleus. So the first point to differentiate the WBCs and the RBCs, you will find according to the size. So RBCs are huge in number, but are somewhat smaller in size than the WBCs. Mainly the large WBCs one, the RBCs are much smaller than the larger WBCs. Now we don't classify all the WBCs and large or small. Lymphocytes are found. Some lymphocytes are very large, some lymphocytes are small. So we classify the lymphocytes as large lymphocyte and small lymphocytes. But neutrophils, it will be say just like 1.5 times the size of the WBC. So as you go on looking under the microscope and finding cells, so this experience will come to you, that at least it will be somewhat larger in size than the RBCs. And there are other structural differentiation features, but first you note the size, that this WBC, this is not looking as the structure which are pinky structures like the RBCs. These are these nucleus will be there. So nucleus structure, how to look for, will know just now we will come to that. But these purpley structures, these structures are somewhat larger in size. Neutrophils, eosinophils, basophils, maybe 1.5 times to 2 times the size of the RBCs. Large lymphocytes, again 2 to 2.5 times larger than the small lymphocytes. Large lymphocytes are larger than that also, than the neutrophils, eosinophils, basophils. Small lymphocytes are almost the size of the RBCs, but the color will vary, okay, deep purple color, okay. And monocytes are the largest cells among all these cells. RBCs are larger than, usually larger than the RBCs of course, and all the other types of WBCs. It may be around, this year you are not measuring it. So never, we know theoretical measurements of the RBCs and WBCs. It is expressed in micrometers. We know that theoretically, but during when you are looking under the microscope, you cannot tell any measurement. So that will be a foolish statement to make, because you are not measuring those. You are just comparing the size of the WBCs with the size of the RBCs. Okay. So monocytes are larger cells. You will see the surrounding RBCs, and maybe it is 2 to 3 times the size of the RBCs. Now, how you can easily differentiate, what we told is, what we can easily differentiate is the size of the WBCs, the color of the WBCs, the stain in color. After putting the Leishman stain, we are looking under the microscope, it can be differentiated by the color. These are the WBCs, these are the RBCs. But among the WBCs, you have to identify which are neutrophils, which are lymphocytes, which are eosinophils, which are basophils. So eosinophils, there will be coarse granules. So just all these points are given in the book, but just write down eosinophils, coarse granules. You will find it in the books, but just write down eosinophils, coarse granules, and write the term bilobed spectacle shaped nucleus. Bilobed spectacle shaped nucleus, yes, these days people are calling headphone like structure, but headphone like structure is not a term used in the standard textbooks. It is written bilobed spectacle shaped nucleus. So nucleus, there will be two lobes. Okay. So remember the nucleus of these granulocytes appear lobed after staining with the Leishman stain. These lobes are identifiable under the microscope. When we stain by the Leishman stain or other stains also, there are other stains available. Here we use the Leishman stain. There are the GMs stain, Wright stains. Okay, but here we use the Leishman stain. The lobes are identifiable. In case of eosinophil, it is bilobed and it looks like a spectacle shaped. Okay, bilobed spectacle shaped nucleus with coarse granules. And how do these coarse granules look like? Pinkish, because these granules are stained by eosin, but the nucleus is stained by methylene blue. Okay. In case of neutrophils, it is particularly for the beginners, it is not very easy to identify, although there are fine granules. So neutrophils, you try to identify by the number of lobes. These can be two also, like eosinophil, there are two lobes, it looks like spectacle shape, but neutrophils, you don't see that spectacle shape. Neutrophils maybe two lobes, maybe three lobes. It is up to 7 lobes may be there, maybe 4 lobes, maybe 5 lobes, and there are fine granules. And what you consider while identifying the size? Neutrophils, eosinophils almost the same size, but eosinophils spectacle shaped nucleus and bilobed. Neutrophils may be more than 2 lobes, maybe 2 lobed or more than that, up to 7 lobes. Right? Again, basophils, granules will be there. Basophils, granules will be there, lobes will be there, few lobes will be there. But basophils, at least as beginners, don't bother too much. Basophils are rarely found. Basophils, so you have to do the differential leukocyte count. Differential leukocyte count is counting this number of different types of leukocytes, and you have to express it in a percentage. So if you say count 10 or 20 leukocytes, after counting, you will find that none of them are basophils. Mostly none of them are basophils. If you count 100, 200, then 1 or 2 may come. Okay. So basophils are very few in number. At least as beginners, it is better to not comment any cell as basophil. So practically you comment neutrophil or eosinophil or lymphocyte or monocyte. Okay. So my suggestion will not to comment any cell, this is a basophil. If you comment, you have to stand why you are saying that. Neutrophils you can stand that see we have found these 3 lobes, the granules are fine. This is almost slightly greater than the size of the RBCs, and there are so many lobes, there are fine granules. Eosinophil, bilobed spectacle shaped nucleus you can see, and the coarse granules, coarse pinkish granules. But basophil, it can be identified, but these are so few in number, it is better not to comment. Okay, basophils are very few in number. Counting 10, 20, 30 cells, maybe there are no basophils. Counting more, there will be 1 or 2. So out of 100, maybe 1. So theoretically, we tell basophils are out of 100, 0 to 1. So it may be 0 also. So counting 500,000 cells, then there may be a few basophils. So it is better not to comment about that this is a basophil. It is either neutrophil or eosinophil if there are granules. And in case of lymphocytes and monocytes, there are no granules. Okay. In lymphocytes, it is easy to identify. In lymphocytes, the cell membrane is identifiable, and the nucleus will cover almost the whole of the cell. It will be a rounded nucleus covering almost the whole of the cell, and it is stained that deep purple it is deep purple in color. So if the nucleus is almost whole of the cell, this is the cytoplasm is just surrounding it. This is called this is described as a thin rim of cytoplasm outside the surrounding the nucleus. Thin rim of cytoplasm. Rim is this rounded. So this rounded portion, thin rim of cytoplasm. Okay, so lymphocytes are rather easy to identify. This thin rim of cytoplasm outside the nucleus, and the round nucleus covers almost the whole of the cell. Now these lymphocytes, some lymphocytes are, just by looking at the size, you may comment as large lymphocytes or small lymphocytes. Small lymphocytes are more or less the size of the RBCs, maybe a little larger, but large lymphocytes are much larger. If you compare with the surrounding RBCs, it is easy to comment whether it is a large one or a small one. And monocytes are larger than the large lymphocytes also. Monocytes are larger cells than the large lymphocytes also. Okay. Now again you will compare it with the RBCs, or if you have found some neutrophils already, if you have found one or two neutrophils, one or two eosinophils, okay, few neutrophils. Neutrophils are the most abundant among the WBCs. Okay, you have found few lymphocytes, you have found eosinophils, then you will find among all those, monocytes are the largest ones, and again it is easy to identify because it is a typical like the shaped nucleus. This is called a bean shaped or a kidney shaped nucleus, and this lies towards one end of the cell. So like in lymphocytes, it is just a thin rim of cytoplasm outside a round nucleus which is purple stained. In case of the monocytes, it is a bean shaped or a kidney shaped nucleus, and an abundant cytoplasm like in the other part of the cell, and the nucleus is towards one end. This is a bean shaped, and the shape is also typical. It is bean shaped or kidney shaped and easy to identify, plus it is much larger in size than the RBCs and somewhat larger than the other WBCs also. So these types of WBCs you have to identify. Okay. Now comes, after you have learned to identify the cells, you will have to count them and express it as a percentage of 100, which is about the differential leukocyte count. So we will discuss about how to count and take the result of the differential leukocyte count. You will do it in the next class. For today, now just proceed to the laboratory. Those who are new, try to learn how to stain, okay, how to prepare and stain the blood smear, and identify the cells the few cells under the microscope. This will not, this is not possible to be very efficient with it in one day. Okay, so we will do it in the further classes. We will take about say 2 weeks for this topic.